another example of a trait. Tall, short, your height would be another example of a trait. And so most traits are the result of a number of different genes in our genome. But it's looking at the genetics of a trait like that is hopelessly complicated. So we're going to try and focus on traits where only one gene determines the trait, like eye color. Generally, only one gene determines eye color or hair color or things like that. Certain diseases are only caused by one gene, so they're easy to look at. We will concentrate on those. Bear in mind, there are a whole bunch of traits that are determined by more than one gene. Like nose shape is determined by a whole bunch of genes. Like if you got a nose like mine versus a better looking nose. So,
Question.
yeah.
A little off topic, maybe a little on topic. Eye color. So in the first 6 months of life, it kind of changes.
It kind of changes. And gene expression can change. And eye color is really more complicated than we make it out to be. It's actually determined by melanin. It's not determined by some other pigment, as our discussions would have you think. And it actually is determined by more than one gene sometimes, especially colors like green eyes. And so blue eyes and brown eyes are a little easier to talk about, but that's another example of where it's probably in that what you're talking about is more than one gene involved. So, okay. Many genes are variable. That is, many traits are variable. Let me put it that way. Like eye color, you can have different colored eyes, and so if it's only determined by one gene, it's easy to talk about. And an allele is is a form of a gene. Like you could have, if we're talking about eye color, you could have, there are multiple alleles. You could have the blue allele or the brown allele. Get it? Or same with hair color. And some genes only have one allele, like the genes for enzymes. The enzymes are very specific in their structure, and there's only one allele. If you have a wrong allele, you won't have the enzyme, and you probably will be dead. And you won't care because you're dead. So we're only interested in traits with multiple alleles because they're interesting to talk about. This guy Mendel, who did all the original work, worked with pea plants. And pea plants are a lot simpler to talk about than humans because in the first place they only have 7 chromosomes. And he got lucky and he picked traits that were all on different chromosomes. So they traveled independently of each other, and he was just phenomenally lucky. Like for instance, traits he would look at were tall versus a short plant, or the shape of the seed, or the color of the flower, or something like that. Phenotype is what the organism looks like as a result of what alleles it has. Okay? We all have 2 copies of every gene. We got one from our mom and one from our dad. And so we have 2 genes for eye color and 2 genes for hair color, and things like that. Suppose it's the gene that causes cystic fibrosis, which is only caused by one gene. There are 2 genes for that one too, and you either have a good one or a bad one. Most people have good ones. And things like that. So the genotype is expressed by a couple of letters indicating what particular genes, 2 letters, telling for this particular trait what genes you have, what alleles you have, okay? And we'll see some examples of that. It's always 2 letters. If both letters are the same, you have a homozygous genotype for that trait. If both letters are different, there's only 2 different ways they can be. You're either homozygous or heterozygous. And if you're homozygous, you know what the person's gonna look like, okay, because that's the only alleles they have. If you're heterozygous, then there are more questions to be answered. If you have 2 different alleles, which one is gonna be expressed? Or is it gonna be a sort of a combination, or is there gonna be a contribution from both? See? And so we'll see that in some situations, only the dominant one is expressed, and in other situations, like whether or not your hair is wavy or straight, both alleles make a contribution. So we'll look at different situations. I think I forgot to mention phenotype. Phenotype is just what you look like as a result of what alleles you have. So if your genotype for eye color is one where you have both alleles for blue eyes, your phenotype will be that you'll have blue eyes. It's kind of easy, okay? So there'll be you'll see these on the first page of the quiz in a matching question. Should be pretty easy. Okay, so here's an example. We use capital B for brown eyes because we tend to use the capital uppercase letter for the dominant allele, and brown tends to be dominant over blue, and we use the lowercase for the recessive. So if you have this genotype here, what eye color, what's your phenotype going to be?
Brown.
It'll be brown, won't it? And we know what it'll be here. It'll definitely be brown. What genotype would you have to have blue eyes?
Homozygous blue eyes?
Yeah, homozygous, little b. Homozygous recessive, it's called. This is homozygous dominant. This is heterozygous. So, get the idea? Okay, and so this Mendel was a monk way back in medieval times, and rumor has it that his students were so terrified of him that they often fudged the results to come out to what they knew he wanted to have, because evidently he'd get very angry if it didn't work out the way he wanted it to. So I don't know if that's true or not, but anyway, he's the one that did he did some good work. He's considered the father of genetics. Okay, so each simple trait is determined by its own gene, and a simple trait is one that's only determined by one gene. Okay, traits vary because genes vary. The alternate forms of genes are called alleles. Do all traits have more than one allele?
No.
No, most traits I would say that 80 to 90% of all of our genes are genes for enzymes. Now, we don't want to have any variation in those. We want them just to be the one that works. Okay? And if that enzyme doesn't work, then you can't do whatever chemical reaction that it catalyzes, and you probably would never have been a viable fetus in the first place. Adult organisms have 2 copies of each gene. You got one from your mom, one from your dad. A gamete is produced by a certain kind of cell division. Give me an example of a gamete. You guys know what a gamete is? Sperm or an egg. And gametes are produced by a form of cell division called meiosis that only occurs in the gonads. And it results in the gamete only having 1 copy of each gene. Okay? But that's what you want, because when the 2 gametes get together, then once again, the result has 2 copies of every gene. See, if you only got 1 copy of every gene, the cell is said to be haploid, meaning half the normal complement. If it has 2 copies of every gene, the cell is said to be diploid. So the most common types of cell in our body are diploid. And we have 2 of every chromosome, all 23 chromosomes, we have 2 of each. Got 1 from your mom and 1 from your dad. Okay. So most cells in the body are diploid. What's the only cell that's not diploid? Gametes. If an adult's 2 copies are the same, it will have the trait specified by the allele. If the adult's 2 alleles are different, it will have the trait specified by only 1 of the alleles. But of course, we come to find out that's not always true. But in the one traits he was looking at in pea plants, it was true. You see, with pea plants, you can grow a few thousand of them. And so you have what's called the statistical cross-section, a meaningful number of results, that you can count. So if you've got like 3,000 pea plants and you wind up with, or let's say 4,000, and you wind up with 1,000 of one kind and 3,000 of the other, that's enough pea plants that you can say with some amount of certainty that, well, there's a 25% chance that this thing will happen, and there's a 75% chance that that thing will happen. With humans, can you do that? Humans don't have enough offspring to constitute a statistical cross-section. You can have 2 parents that are carriers for cystic fibrosis, and there's a 25% chance that their kid will have cystic fibrosis. They can have 10 kids and have all 10 with cystic fibrosis, or have none of them with cystic fibrosis. It's like going to Vegas. You know what the odds are. Does that mean that's what you're going to win? Well, if you play statistical cross-section of games, it should be. But no one does, because you can't afford to. And so, but the house experiences a statistical cross-section of games. So the house always knows what it's going to make. See, it always knows exactly what it's going to make at the end of the day. Because there's a statistic, it knows what the odds are, and the odds are always followed if you have a statistical cross-section, a statistical valid number of events. So, okay. So chromosomes are carriers of genes. Humans have 23 pairs of chromosomes. A typical chromosome has about 10,000 genes. And each pair is said to be a homologous pair. That is, it carries genes for the same traits. They know what traits are on what chromosome. That was what the Genome Project was all about. Okay? And like I say, a typical chromosome has upwards of 10,000 genes on it. So they've mapped the whereabouts of all the genes on all the chromosomes. Okay? And these chromosomes have about 100,000 genes, give or take, really more like, I think, about 60,000. You have 22 pairs of non-sex chromosomes and 1 pair of sex chromosomes. The non-sex chromosomes are called autosomes for some reason. So they know here's a pair of homologous chromosomes that carry genes for the same traits. Maybe it's chromosome 7 or chromosome 16 or whatever. An expert can tell by the shape of a chromosome what its number is, and thereby know what traits are on it. Okay? And they know where the location of every gene is. Okay? And you could either have, with both these chromosomes, the alleles could either be the same or be different. Okay? Alleles can also be mutated. In many cases, that mutation will work better And that's one of the foundations of evolution. And in most cases, it won't work at all. And you'll have a non-viable fetus if that's an important gene. If it's something like hair color, it's not, your life doesn't depend on what color your hair is. And so you could wind up with some wild new color of hair that no one else has, you know, and it wouldn't matter to your survival. So mutations happen. As a matter of fact, there's a built-in mechanism for mutation. During DNA replication, about 1% of the DNA is not replicated properly. And so mutations automatically always result to at least 1%. And so that produces, as you look around you, you see that all of us are different. And if you look at young people, they have characteristics that are a little bit different maybe than we have. So the species is always changing. There are two foundations for evolution. Number one, the species is always changing, and number two, natural selection. A particular variation, a particular phenotype may be the most successful one now, but 50 years from now, with our constantly changing environment, it may not be the most successful anymore, but we are constantly changing, and it might be that some new variation will be more successful. And that's what natural selection is all about. Nature selects the most competent, and that, you know, individual, the most competent phenotype. That phenotype will be successful and prevail and multiply and perpetuate its new allele. That just happened by mutation, see? And so mutation is always happening. And so some mutations, like the mutation that causes sickle cell anemia, are not good, but people tend to live to reproductive age with them, so they're carried on anyway, or the one for hemophilia. People live to reproductive age at least, so it's perpetuated. So here is the reason why they used to do amniocentesis, and now what they're doing is chorionic villus sampling, which isn't quite as invasive. The getting some fetal tissue is so that they can get a karyotype. And they can't see individual genes, but they basically, what they're interested in is counting chromosomes. Do you see any problems with this particular karyotype?
There's missing one?
No.
What's the deal on 21?
It's got 3 21s. And this is the kind of thing that they're looking for. This individual will have Down syndrome or as it's the more modern term is trisomy 21. So the reason they do a karyotype, and they have techs who can whip these things out really fast, they catch a cell that's in the middle of cell division, and they can pick these chromosomes according to type, and you know, see if you have the proper number of each one. So, is this going to be a male or female?
Male.
Male. Okay, so if you look at the hierarchy of structure in a chromosome, okay, here is looking at what they look like if you're standing far away, but if you look closer, you'll see that it's actually a big coil, okay? And if you look within one of those coils, it will look like this, okay? And each one of those filaments will be made out of chromatin. Those little filaments there will be made out of chromatin, which is what we see here. And if you look more closely at that chromatin, you'll see that it's DNA, a strand of DNA wrapped around these proteins called histones. And the histones have a lot to do with what genes are expressed and what genes aren't. A gene is a segment along the length of a DNA molecule. DNA molecules are almost infinitely long, continuous molecules, okay? And if you have a gene that codes for a very large protein, then the amount of DNA that's required in that gene will be a long segment. If it codes for a gene or for a protein that's a little short one, with maybe only 5 amino acids, then it'll be a short little segment of DNA. Okay? When that double helix unravels, the gene is expressed. Most genes are asleep most of the time. Only about 1% of the genes in the cell are expressed, because after all, the cell contains all the genes to be any cell in the body, doesn't it? Because all the cells in our body came from that first cell, and that first cell had to have all the genes necessary to be a brain cell or a bone cell or a liver cell. But of course, all cells aren't. The cells are only one kind of cell, so they will only express the genes that it takes to be that kind of cell. And they won't even express all of those all the time, just the ones that are needed. Okay. So stages of cell division, it's pretty easy to break a cell apart. Like you could split up, these cell membranes are liquidy, so you could just kind of pull those into 2 different cells and split up the mitochondria and that kind of thing, and split up most of the other little organelles in there. But the one thing that's tricky is splitting up the genetic material. So when we think of the stages of cell division, we think of the stages of divvying up the genetic material. You want the proper genetic material to go to the 2 daughter cells. You want it to be split equally, so that each daughter cell gets 1 copy, gets its number of copies that it needs to have of the gene for each trait. Okay? And you definitely want, of each pair of homologous chromosomes, you want 1 chromosome to go in each direction. Big mistakes, that's what leads to Down syndrome, is when 2 chromosomes go one way and no chromosomes go the other way. Okay? So prophase, when you look at the nucleus of a cell, normally it can't see anything in there. What's going on in there, and this is what it looks like most of the time, is you can't see anything. It's because the DNA is very busy replicating. Okay? And finally, when all the DNA is replicated, then the cell, you have to double the genetic material in order to split it in half. Okay? And so once all the DNA is doubled, then we're ready to split it in half. And so prophase is the first phase of cell division. It's where the chromosomes actually take shape. Pretty amazing. And they take on these distinctive shapes that allow you to say, oh, that's a chromosome 6, or that's a chromosome 13. Metaphase is where they line it down the middle in a very orderly fashion, like a bunch of little soldiers, so they can keep. Anaphase is when they're pulled apart in a very orderly fashion. And then once they're pulled apart, they go their separate ways, and the membrane forms in between the 2 ranks that have gone their separate ways, and all of a sudden you've got 2 separate cells, daughter cells. Okay? And telophase is where the chromosomes in the daughter cells lose their shape. And once again, the nucleus is just an amorphous blob of nothing. Okay? So we're interested in the metaphase when they line up. Okay? There are 2 different kinds of cell division where you have different metaphase lineups. Okay? They can either line up single file, like this, where the 2 chromosome 1s, 2 chromosome 2s, like this. And this is what's gonna happen if your cell division is mitosis. There's only one way that you can line these things up, single file, isn't there? There's only one way you can line them up. So you may have been puzzled at some point in your life about why sometimes chromosomes are shown as these, what I call bow ties, little double lines, and sometimes they're shown as single lines. Why can I call that a chromosome, and then in the same breath, call that a chromosome? Okay, people do it. Well, what happens just after the bow ties are pulled apart, what we're really doing is pulling the halves of the bow tie apart. The halves of the bow tie are identical. That's what we've been doing when we've been replicating the DNA. We took these single chromosomes and turned them into a bow tie with 2 chromatids, and the 2 chromatids, the 2 halves of the chromosome, are identical. Each one, if you pull them apart, you'd have 2 chromosomes. There's kind of a semantical problem with that, seems to me. If I called this a chromosome and called those separate chromosomes, but nonetheless, that's what they do. And so that's what's going on, is the single ones are turning into double ones. You can tell if the cell just divided if the chromosomes you're looking at are single ones. You know it just underwent cell division. If all of them are bow ties, you know it's getting ready to undergo cell division, but it hasn't done it yet. Okay? So you'll see them in different forms. So mitosis is the type of cell division that goes on everywhere in the body. And there's only one way these guys can line up single file. And if you pull them apart, you'll have 2 single file ranks of chromosomes that are identical to each other. 2 1s in a row, 2 2s in a row. So the daughter cells are identical to the parent cell. But that's what you'd expect, because we all began with one cell called the zygote, fertilized egg. Okay? And we pull them apart, and that one cell undergoes mitosis to produce 2 identical cells. Then the 2 identical cells give rise to 4, and then 8, and 16, 32, 64. That type of thing. But mitosis is what goes on everywhere in our body except the gonads. And all the cells are identical in our body. Their pattern of gene expression isn't identical, but the genes they possess are all the same. Okay? In meiosis, okay, this is what goes on in the gonads. And instead of lining up single file, the chromosomes line up side by side, where the 2 1s are side by side, the 2 2s are side by side, and like this. And you see, if they line up side by side, the picture I've drawn here, you can't tell which one you got from your mom and which one you got from your dad. Okay? But if I look at it here, it took me a long time to draw this. But you can see I have mom and dad, and the only constraint is that the 2 1s got to be up here, but whether the mom is on the left or the dad is on the left is variable. So they can flip either way. So you see, if I only had 2 pairs of chromosomes in an organism, okay, there would only be 2 ways they could flip, right? Okay, so you could have 2 possible metaphase lineups. But every time you introduce a new chromosome pair, you double the number of possibilities that you can have. Okay? So by the time you get to 23 pairs of chromosomes, it turns out that there are over 4 million different metaphase lineups you can have. And there's nothing that is going to determine whether the mom's on the left or the mom's on the right. It happens. It happens so quick, and You know, and so if you've got a bunch of cells, like a typical male's making 300 million sperm a day, there could be every single one of those 4 million metaphase lineups could be occurring. Probably is, if you got 300 million tries. Well, not, if you got 300 million tries, yeah, you could easily make just 4 million variations. And so you see, when you pull those apart, you know, we get different individuals just from pulling those apart. And for every metaphase, different metaphase lineup, you can get a different, an individual with different genetic information. From just 2 original parents, you could get 4.5 million different individuals with different genetic information. That's why the sperm and the eggs have to be protected from our immune system, because our immune system would recognize them as foreign and attack them if they weren't isolated from our immune system. So it's because meiosis gives rise to different genetic individuals. Okay? And so that's why virtually every sperm a guy produces is different. And a female's born with about 1 million eggs, and they're probably all different from each other too. Okay? They're mostly different. And so, you know, it's just whichever one gets fertilized, if that's the person you get. So from just 2 parents, look at the huge distribution of different genetic individuals you can get. It's quite amazing. Meiosis can give rise to all these different possibilities. So gene shuffling is what a lot of people call that the mom and dad chromosome can be flipped either way. If you think about it, from what I said verbally, you can easily derive this little formula that I derived. And basically the number of permutations you can have in your metaphase lineup is 2 to the n-1, where n is the number of chromosome pairs. So if you have 2 chromosome pairs, there's only 2 different ways you can flip them. So 2 to the 2 minus 1 is 2 to the 1, isn't it? And 2 to the 1 is 2. 2 different ways you can flip 2 chromosomes, right? You introduce another one, and it's 2 to the n-1, then you got 3, and 2 to the 2 is 4. There's 4 different ways you can, 4 different metaphase lineups you can have. So I think I got a picture. Well, I don't, but meiosis, this just shows meiosis 2, where, remember meiosis 1, you just got